Okay, let's talk about active transport. Active transport, we said in the last video, is going to require ATP. It is also always going to require a carrier or a transport or channel protein. Okay, so it's always going to require a protein to help it get across and ATP. Now, when we think about this, when we talk about going down the concentration gradient, it's like we're going in the flow of the river, right? The river kind of takes us in its flow, but if we want to go against the concentration gradient, then we have to sort of pump. If we think of like a sodium-potassium pump, which I'll mention in a moment, we want to put energy into it to turn around and go against the flow of the river. So if we're moving against the flow of the river, then we need ATP to do it. We need to add energy to the system, and so that's what makes it active transport. I think the best example of active transport is sodium-potassium pump. Sodium is, when we open gates, sodium is going to move in the cell because it mostly lives outside. Potassium is going to move out of the cell. They're moving down their concentration gradients. So, we don't want the electrolytes, which sodium and potassium are both minerals and they're also considered electrolytes. We don't want there to be an electrolyte imbalance because it's necessary for nerve conduction and muscle contraction. So we have to rebalance that. We've got a pump called the sodium-potassium pump. And what that's going to do is these proteins, with the help of energy, are going to pump sodium back outside the cell and potassium back into the cell so we can restore our ionic concentrations. Sodium and potassium being ions, okay? I think I mentioned in a previous video that cations are positive, and so sodium and potassium are both cations. And so we want to restore the ionic concentrations, okay? All right, so that is a form of active transport. What's really interesting about that is when we move sodium, we pump it back across the membrane, we change a couple of things. They generate something called secondary active transport. Because when I move sodium over, I've done what? Well, it's a cation, it's positive. So if I move sodium to one side of the membrane, what did I essentially do? I've changed the charge, right? I've made it one more positive on this side, or one less negative on this side, right? So I've changed the charge, and opposite charges attract, so that can draw some anions with it as we move sodium to one side of that membrane. Okay. The other thing it does is it increases the concentration on this side of the membrane. So we change the chemical gradient or the concentration gradient. And what did we say in the last video about osmosis? If we have a higher concentration, that means we have a higher osmolality, and the higher osmolality is where water wants to go to. So if we increase the osmolality on this side, water wants to flow to that side. Remember, water always wants to dilute, so water is going to move to the more concentrated particle side. And so we have that secondary active transport taking place that does not require additional ATP, by the way. By moving sodium over, we change electrical and concentration gradients, and that is going to help stimulate the movement of some other things across the membrane. Okay, a couple of other active types of transport. We have something called exocytosis. Exocytosis, EX, think exit. Exocytosis is going to be this. If we've got a cell membrane, okay, we're going to put a, let's call it a hormone, we'll say a hormone would be a good example. We're going to put it in a little vesicle, okay, and then that hormone is going to bind to the vesicle, the vesicle is going to bind to the cell membrane, and then it is going to release, as it binds to the cell membrane, it becomes part of the cell membrane, we're going to release that particle, okay, or that chemical or that hormone to the outside. That's exocytosis, okay? So the vesicle that contains the neurotransmitter or the hormone is going to bind to the cell membrane, and then it's going to evaginate, so to speak, where it exits or causes an exit of that particular chemical, all right? We have endocytosis. In endocytosis, we have a couple of types. One type of endocytosis is called pinocytosis. And here's what happens. You have now this is for smaller particles, things like that. So you're going to have some nutrients, things like that. You're going to have this particle contact the cell membrane. The cell membrane is going to invaginate, okay? And what's going to happen is it's going to pinch off a vesicle. So the cell membrane is going to invaginate, pinch off a vesicle that's now going to contain that particle. And now we've moved it from the outside, as that cell membrane invaginated, we moved it from the outside to the inside. That's pinocytosis when it's smaller particles. When it's larger particles, a good example of this would be like a macrophage engulfing a bacterium. Okay, bacterium is singular for bacteria. And so what will happen is, again, a larger particle, we're going to have the invagination of the cell membrane that's going to, again, move the particle to the inside, and then we do some other stuff with that. But the important part we're talking about here is the phagocytosis, and the phagocytosis is going to, again, invaginate the cell membrane to get larger particles in to the inside from the outside. Okay. Now again, when we're talking about primary active transport, when we're talking about exocytosis, when we're talking about endocytosis, we have to add energy to the system. These are energy systems. Okay. Another thing that we can just throw this little wrinkle in there, and we'll kind of end on this note, is sometimes it's called receptor-mediated endocytosis. So what does that mean? Well, it sounds fancy, but really all it means is there's a receptor on the cell membrane, we need the binding of a chemical, and that binding of the chemical is what's going to stimulate the invagination. It's going to cause the vesicle to move to the inside, or the piece of the cell membrane to make a vesicle to move to the inside of the cell membrane. So it's endocytosis, just the same, we're moving from the outside to the inside. Endo means within, exo means without or outside, and we can think of it as exit. But we're going to have to add energy to it. With the receptor mediated, we're also going to have to bind a chemical to a protein receptor to cause the membrane to actually invaginate and get that endocytosis process happening. If you have any questions, let me know. Stay strong. I'll see you next time.